Everyone. It's 1 o'clock and we're going to go ahead and get started. We'll give just another minute or two for people to join in. So thank you everyone who's logged on thus far. If you want to go ahead and change your view option to fit to window, that will give you the best viewing. And if you want to use the Q&A button at the bottom of your screen for questions, why don't we go ahead and test that out a little bit. So if everyone who's on can go ahead and let us know where you're viewing from. This has been an interesting adventure to see how far reaching this webinar has gotten. Thomas from New York City. We have about 20 people on the call thus far. Robert from Fort Myers. Hi to Mike from his old stomping grounds in Ashland, Oregon. Sunny Yardley, Pennsylvania. Hi, John. Redwood City, California. Hey, Ron. Great. So it's 1:02. We'll go ahead and get started. Hello, everyone, and welcome to the second event in our MetroFlow Corporate Member Launch Seminar Series. I'm Christina DeCoste, Director of the Flow Cytometry Resource Facility at Princeton University and President of MetroFlow, the New York, New Jersey Flow Cytometry Users Group. Since we're unfortunately not able to meet in person during this time, the 15 members of the MetroFlow Steering Committee have developed this launch seminar series as a vehicle for our corporate members to reach our MetroFlow audience via this virtual platform. This session is the second of several to follow throughout the remainder of the summer. And as a disclaimer, MetroFlow remains neutral and in no way endorses any one vendor or their offerings over another. All corporate members have the opportunity to submit a proposal to present in this launch seminar series. And the purpose of these events is to showcase the innovations in the field that we can all take advantage of to better support our scientific and research endeavors. We hope that all of you will be able to take what you learn from this seminar series back to your labs and that the information will be helpful to all who are watching today and in future seminars. Please use the chat function to report any technical difficulties and the Q&A button at the bottom of your screen to submit questions. And before we get started, I'd like to thank today's speakers, as well as you, our MetroFlow members and extended audience, for your time and participation. And with that, I'd like to introduce our speakers, Mike Stedniski and Seddon Thomas of Phitonex.
Awesome. Thank you, Tina. Thanks so much.
Okay, Mike, you can just go ahead and share your screen.
That's what I'm doing. Here we go. Great. Thanks for showing up today. We definitely have some fun planned. Only a couple new floors and maybe a new laser line. So the way that I like to think about launch is also staging a launch, right? Very similar to the way that we put Bob and Doug up onto the ISS. We'll bring this back a little bit, but how do you think around having a first and a second stage when you do have a large number of floors? How do we like to think about what the reusable components of that are? And then also, how are you going to bring your friends along, right? And so just put a pin in that for now. We'll come back to this concept a little bit later on. We think about how do you actually think around launching new floors. The slide I've started, honestly, every webinar that we've done over the past, several months has been looking at how many floors are we looking at over what year have they been achieved. And we'll come back to this several times today. But this is all leaning on Mario's law for flow, right? A law that will break quite liberally today, where the number of colors doubles every 10 years. I would note that when we really look at this, right, that it's really been slowing down, particularly as of late, right? When we look into the late 2010s, we don't really get a lot of development of new floors. And arguably a lot of the new floors, even that we've seen in the past several months, have honestly been derivatives of some of the historical floors that we've seen in the field, be they PE or APC, right? I'm talking about new floors, right? New to the world floors. So earlier on in 2020, we released the first 6 blue, yellow, green labels. Had the opportunity to present at MetroFlow some of the work that we've done in using that to get up to 40 colors in flow. And today, just to cut right to it, we'll be releasing 13 new fluors. So we actually did this last week, but now we have 19 NovaFluors. So we're heading towards the full vertical in 2020, at least in the first half of this. And when I say launch, that does have a pretty important note for us. That means that we've taken these fluors, developed them through a process that I'll show you, conjugate them to antibodies, and also run them in flow cytometry. As I'll describe, flow is a critical part of everything that we do in technology development, and I'll describe how we like to think about that, in the context of current labels and what it means to treat fluorescence like a design problem. So I think that's going to be a lot of fun as well, and hopefully adds in the understanding and giving a little bit of a preview too of what else we have to work on. The first 10 or so fluorophores, took 20 some years to do. The next 19 colors took 24 years. And we've done 19 labels in 6 months. So I'm really excited to get into this today. I'll show you some profiles too of how these compare to some of the current dyes. And we'll talk a little bit too around dye history, honestly, just because it's fun. And so we'll start here with the ending. What are the fluors we'll be talking about today? So we've added an additional 5 fluors off of the blue. Again, I'll show you some performance characteristics on this as a flyer. Really, we've gone in and created some new blue dyes that are really good FTSE replacement, better on separation index, lower on spread. We're also introducing a brand new concept into flow cytometry, which is actually the ability to choose your separation index or choose your brightness, which I think has a really interesting impact on panel design, which we'll talk a little bit about. We're releasing 4 new labels in the yellow, very, very bright labels here, as you would expect for things that were going in and replacing PE conjugates. And then the new laser line that we're rolling out, really we thought a lot around, well, what are historical fluors that we need to go in and really think around engineering for lower spread and clean fluorescence? And the answer was APC. We've leaned on APC for a very long time, as we'll discuss, and we've got 4 new labels that are coming off of the red there. And you can also see now that we can hit gaps of about 10 nanometers or even 10 to 20 nanometers and really get clean fluorescence there. So I'll show you profiles for each one of these labels as well. I think a really important concept that we're going to roll out today, and it took us a while to sort of just like wrap our head around how do we best communicate this, is we like to think around our labels now as light bulbs, right? And the way we name these is here's the excitation laser blue, and here's its emission peak 610, right? So that gives you a sense of, okay, well, that's the compatibility. That's how I can use it on my flow cytometer. And then we thought, well, okay, it's really wattage that we're talking about. You go into a dark room, you need a high wattage bulb. You've got very low antigen density. You need more separation index to pull those up. But similarly, if you have a very high antigen density marker, right, you don't want the brightest thing on there, right? You'd really like to pull that down a little bit so it doesn't contribute spread in other parts of your panel. And that's a concept we're introducing today. So two of our fluors actually have different separation indices that you can choose. And these are quite literally their separation index. So think about these as wattage. And so I'll show you some data on this in a moment. So the way I've organized up today is I'll do a brief introduction to how do we like to think around fluorescence as a design problem. Then we'll go a little bit deeper into that. What's the technology platform? How do we think around that? Then we'll talk around the new labels, right? That part will move rather quickly because we'll just sort of go through the profiles of each label. And then the other part of this is Set, and she's going to speak to new applications, talk around how we think about leveraging these in panel design. We'll talk a little bit about intracellular. We'll talk about stability. And so, how do these work in the real world? And there's some new data to share there, which I think is really super exciting. And then we'll come back around and answer the question of like, how is this working? How are you staging this? How do we get access to these? So the first part of this is how do you design a fluorescent label? And so to that end, I'll start with what are the these are quite literally requirements that we design on, right? Very similar to the way you design software, which is the way I like thinking around it. And the first one is that these labels have to be bright and clean. And so I'll show you this for the first 6 labels that we've done that we're actually going to be moving on to antibody content. And those will be available actually for pre-ordering now and for purchase at the end of July. But we really mean that we're independently exciting different fluorophores and then stacking laser lines. So I'll mention that in a moment when I show you our first 6. The other part of this is consistent and stable, right? So when we were at Cyto last year, people said, it's great, new colors. How does it work in the real world? So I'll show you both our lot-to-lot data today. I will also mention, and actually this is what you're seeing here, our labels are actually bench stable across the entire spectrum for at least 3 weeks is the longest we've tested. I'll also show you some data that they're bench stable and fixative stable, right, when they're in conjugated format. And I think that's something that's really exciting when we think around the current working environment. The other part of this is something that I mentioned, which is this notion of digital fluorescence, which we can use honestly in the context of antigen density, but also can use to say, hey, we really need to tune the brightness up of a label to make sure that it competes well and provides separation, for instance, that's comparable to or better than what we have in our current fluorophores. How do we do any of this? So we take small organic dyes and we bind them onto these single-stranded oligonucleotides. And then we can assemble them into this cruciform structure. And this cruciform structure provides us a platform where we can really engineer, right, the excitation and emission and actually change the geometry of the way that these component dyes interact with one another, right? So we are relying upon fret, right, because we're actually transferring photons in between the different component dyes. But these are not, in any way, shape, or form, a tandem dye, right? Not in terms of stability, which I'll show you, and we'll compare to tandems, and not in terms of the way that you actually use them in the real world. You do not need this particular tandem on this particular antibody, so the fluorescence is different to compensate it. You could use CD4, you could use some high antigen density marker to perform all the compensation. The other parts that are really nice about having a platform like this are that I can also tune the brightness of this. I have 4 different clusters, these arms of this cruciform, that I can tune up and down the brightness. Again, something we'll discuss in the context of some of these labels, is I'll show you profiles of labels that we don't think are bright enough. We can immediately go in and stoichiometrically tune their brightness. 2 other parts that I think are really fun about this. One, I get the flexibility of having an oligo-based platform. The way that we conjugate these to antibodies is we actually hang a single strand of DNA off at the end of this phyton, which enables us to conjugate it to antibodies, right? And we'll discuss what that means. I'll actually share some recent customer reviews that have come out just in the past couple of days about how they're leveraging our conjugation kit, what they think about that. And the other part is that we're relying upon oligo manufacturing to make all of these. So we've been very open in sharing our lot-to-lot data. And I think you'll be hopefully very impressed by the fact that we're really aiming to make lot-to-lot and consistencies in these dyes, just in any kind of dye, a thing of the past. What do we mean by clean and bright, right? And so here I'm just looking at the profile of PE Dazzle. And you can see PE Dazzle is just promiscuously excited, right off the 488 line, the 532, and the 561. This really applies for almost all PE conjugates, right? And we'll talk about the character of multiple cross-exciting dyes in a minute when we think around this as an engineering problem. And what we did with our first set of labels is really separate this and say, what if we could use the blue and the yellow independently? Right? And we see this historically in our instruments. This is why a Fortessa, for instance, has a trigon off of it. This is why people have 4 laser Aurora instruments. It's because we haven't done anything in the blue because we've never been able to separate the excitation of those lines. And that's exactly what we've done. We've got dyes that are independently excited off the 488 and 561 lines. That's exactly what our first set of 6 labels was. We have 3 off of the blue laser and 3 off of the yellow. We've shown that you can actually go in and now stack. If you remove PE out of a panel, you can actually go in and now stack those lasers, either on a conventional cytometer. We've also shown in some of the 40 color work that we've done that you can literally drop these in and in a plug and play basis and up the number of questions you can ask on an Aurora. What does this look like, and why is the stacking, why is bright and clean matter? And something that I've seen huge amounts of data over the past several years. And this is the thing that gets me the most excited. It's the simplest plot, but I think it's one of the most impactful. And what we've done here is take CD4 single stain controls with these first 6 labels. And then we've compensated them and just merged them, concatenated them. And what you would expect to see, particularly after compensations, you'd expect to see this trumpet effect. You would expect to see spillover spreading, particularly where there are these adjacent channels, right? I mean, we're talking about emission of 610 and 530. And what you don't see is what's interesting. You do not see appreciable spread at all from these labels when you look at it in this format. And I think it's really interesting. And so how do these, when we did our first testing, we got 2 of these labels to a lab at the NIH. We just said, look, really, how did these compare to what's out there now? And so the first one that they were able to take a look at, and I think it provides a useful framework for how do we think around dye comparisons, was our NovaBlue 610 dye. Ran this on a facsimile. We're looking at all the detectors that are across the labeled laser lines here. And you can see, for instance, this NovaBlue dye isn't as bright, although we'll talk about what brightness means in a moment when we talk about separation index, but it's incredibly clean emitting, right? We have nothing in the violet. It's silent in the violet. It's silent in the UV. Emits a lot less in the green. And this is what we're engineering for, right? Is to have this characteristic of not contributing spread, of not emitting into these other channels. Similarly, we saw a similar performance off of this Nova Yellow dye, which is really a star performer. And you'll see the level of brightness that we're bringing into the yellow is really impressive. In this case, we compared this Nova Yellow 610 dye to Alexa 694, this PE CF594, aka Dazzle, and the ECD dye. Brighter than Alexa, nearly as bright as this PE conjugate, and brighter than ECD dye. But again, notably, very quiet in the violet and the UV, right? And so very, very interesting results for us that sort of let us started leading on us on this journey of, if we can get lower spread and lower cross excitation, that's really interesting. And so how do we do any of that? How do you design fluorescent labels? So the first thing to think about is that, and our CTO Craig kind of pointed this out to me. He said, look, really, there are only 2 types of labels in the world, right? And I thought, well, that's interesting. And the first type of label is one that's narrowly excited, right? It's like something like BB510, but it has the characteristic of having a large amount of spread. Or the other type of label that we have, something like the PE conjugates or some of the other legacy dyes like PER-CP, is that they might be cleanly emitting, but they're cross-excited by multiple lasers, right? And so that you can fit all of these labels into either of these two buckets. As I've shown you, we think about this as more, how do you stack and how do you leverage all of the capacity, all the detectors in a given instrument? And that's exactly what we did with our first 6, right? You can see we're kind of spacing them out into what that might look like on an emission basis. But how does that actually done? And how do we do any of that? And the point is we actually think around fluorescence, again, as a design problem, or even you can think about this if you're in the drug development world, almost like we're looking at candidates, because that's exactly the way this works. We actually design label candidates, we measure their spectra, we test them in flow up front, and then we select them and manufacture that. And I'll show you why that's important in a moment. But we actually take different candidates here, and we actually do something called spectral sculpting. And what that really means is we're choosing candidates that have performance that fits our requirements for what it means for cross-excitation and for spread contribution. You can see, for instance, that if we get a really wide emission like what we've got in blue, or we get shoulders like what we have off of this red candidate, we kick them out, and they're not allowed, right? They're not allowed at the party. And the point too here to make is that we're not designing just one fluorophore at a time. We're designing for the entire spectrum, right? Which is also why we're able to do so many at a time. Then we look at them in flow. And again, what we're looking for is this characteristic of being brightest in the primary detector and nearly dimmest in the secondary, right? We want them to be incredibly cleanly emitting, right? Now that we've engineered for that. And then we select a candidate. When we have a candidate, then we actually move that and transfer that off into manufacturing. And the important part about this all is that if you think about all the things we're talking about, more than 90% of every single one of these fluorescent labels is DNA, right? And so we're relying upon very standard manufacturing to get this done. And the other point that I mentioned earlier is that our lot-to-lot consistency is really, honestly, pretty impressive. So what I'm comparing here is 2 different batches, 2 different manufacturing lots of our first 6 labels off of the yellow and off of the blue. Our spec is to always be under 5% lot-to-lot variability, although you're seeing that we are actually exceeding that when we look at these 2 lots in manufacturing. We also test this by flow cytometry, right? When we actually compare these lots, we do not see any difference in flow, which I'll show you in a moment. And the other thing is we've sent these to an outside lab, and they don't see any difference in the staining indices in between our lots, right? So the goal there, which is pretty bold, is to try and make lot-to-lot inconsistency in labels a thing of the past. So the label you order now or 5 years from now is exactly the same. This is looking at that same lot-to-lot comparison in flow cytometry, and we just do not see differences in the lot-to-lot. And I think this is really important for us. As you can imagine, just like you were ordering large numbers of antibodies, large numbers of dyes, and we do see lot-to-lot variability. We really want to make that and put that to bed. So what do the new labels look like, right? And what are we rolling out? And I also want to roll up some history into this to sort of give us some context of, well, what have we done? What as a field, what have we done in the blue? What have we done in the yellow? What have we done in the red? And so NovaBlue, we're coming out with a number of these different NovaBlue dyes, also rolling out this concept of digital fluorescence where you can change the separation index. But I thought it was fun and honestly, it was just fun to do, is go back and look at the history of what have we done off the blue? And the answer is, well, honestly, not much after really FITSI, right? It's a derivative of fluorescent, was first synthesized in the late 19th century, first described in 42 in life science, conjugated antibodies in 1970, which is when it appears here. And then really, once you get the advent of PE in 1982, you have this long drought in the blue. And as we've discussed, this obviously impacts instrument design where we just don't have much going on off of the blue. It's our consistent joke in the lab. Why don't you put an octagon on all of the X and 4 testers that are out there rather than a trigon? Because we can just go stack those detector arrays. And as I've shown you, the brilliant blues certainly came out. They do have this characteristic of being quite, if you will, loud in the violet, right? And being cross-excited off of that. We, as I mentioned, have these 3 current blue dyes, again, which we're putting on conjugated antibodies now. And we've come out with a whole set of new blue labels, and I'll take you through what do those look like on a profile basis. To profile a dye in the context of what I mentioned, which we've conjugated each one of these to CD4, run them in flow cytometry, compare them to current dyes. We'll just discuss what's their name, also give some guidance on what their compatibility looks like. We pulled 30 plus configurations, right, from universities that have shared these configurations. I would take that as general guidance. Generally speaking, all of these are going to work on FX Symphony or an Aurora, and then it really depends on your detector arrays, obviously, for every instrument. The other thing that we'll show these ribbon plots off of the Cytec Aurora, which is how we've characterized our labels as well as done the performance characteristics, right? Showing the primary detector and noting that, everywhere else you see fluorescence is introducing spread. There's certainly a signature, but if you think about an ideal dye, it really only emits in that one bandpass very narrowly and then doesn't emit anywhere else. We'll also quantify spread. I know if you've ever looked at these dye selection guides, they certainly talk around relative brightness, which is great, but then you analyze your data and you see how much spread is in it, right? So we'll quantify spread as well. We'll stack our labels up against what other labels are showing in terms of spread. And that's a pretty high bar to meet because we're quantifying spread on a 64 detector instrument. So it'll get interesting, but it's also, us being transparent about what the spread cost of doing business is for any of the fluorescent labels. We'll also quantify relative brightness. The way that we're doing that is looking at separation index. We've been pushed really to look at this. And I think it's really an important note that our customers have pushed us to look at, which is that you really want to make sure that, it's great if you have the brightest label, but if you're also dragging up negative, then that doesn't help anybody because you can't separate the biological populations. So what we will look at, we'll count for the 84th percentile of the negative, and we'll actually look at the separation index. And in this case, you're looking at these peaks, right? The separation index of the blue would be much better than that of the green, even though they're similarly bright, right? And so that's the way we'll look at brightness as well. Again, we'll quantify that or classify that, I should say, for our labels and for the labels to which we compare. Here's what a dye profile slide looks for us. And we've got all of this. We're posting this up on the website as well. Actually, all of these profiles are up on the website already. And so I will go rather quickly through this and just give you the high level, why did we make each one of these labels and how does it compare? So NovaBlue 510 really intended, honestly, to go in there and replace FITC off of most instruments. It's actually got a better separation index than FITC and very, very low spread, which is honestly a FITC-like behavior. And so an interesting dye, I think, arguably also a great competitor up against something like a Brilliant Blue 515, which is very, very bright, but has honestly an enormous amount of spread that it contributes. NovaBlue 555, again, another one that, depending on the instrument configuration you have, can go in there and replace FITC or AF 532. Another really interesting dye in terms of it having a really decent amount of brightness, honestly, but a spread contribution that's essentially zero. NovaBlue 585, honestly, obviously a little bit further out. There's really nothing that fits into this detector right now, right? It's against PE, which doesn't make much sense because PE is yellow excited. You'll note here, right, we have a very low spreading dye, but it's also not that bright. This is a great example of a dye that we're actually tuning up the brightness. So we've got this on one of the four arms of the cruciform, and we're actually tuning that up. So we expect a separation index that'll come in at about 16 to 20 as we tune that up stoichiometrically and make some other optimizations. So it's a very interesting label in the sense that it's a brand new to world, hitting essentially a detector that doesn't have anything in it now, thanks to PE honestly having very sharp elbows and pushing everything out of the blue. So I think a really interesting label that, again, should have very low spread contribution as well. The other notion that I've mentioned already is this notion that we can bring digital fluorescence to bear, right? We've done this. We're going to do this on the label I just mentioned in terms of tuning up its brightness. We've shown our ability to actually stoichiometrically tune brightness on the platform. And that stoichiometric tuning of brightness actually quite literally tunes the separation index. And I think this is really cool to think about. Again, we think back to our antigen density and saying, I've got a high antigen density. Okay, now I need to go for a lower brightness. The other thing that a number of customers have pointed out to us is, if I go and design a panel and I've somehow have missed that, right, where I've actually got lower antigen density, I need that to be brighter. When you do that with fixed brightness fluorophores, you need to go and re-engineer the whole panel. With these, you run the panel and you say, well, I really want that label to be brighter. Okay, just move that one up, right, and get a better separation index for that label. And I think this is a cool, really interesting and cool concept for panel design that we will be certainly building out over a larger number of colors. Where we started is actually by doing this off of these two NovaBlue labels. And you can see, you can quite literally choose your separation index for the biological question you're trying to ask, right? And so we've got ones that have 30, 70, 60, and then 120, very, very high separating index. And what's really interesting is when I look at this in histogram basis, because now you could say, well, I really want, I've got a high antigen density label, CD45, right? And I really want to tune the separation index of that down, contribute less spread into my panel. Right, or I want a higher brightness thing because I've got low antigen density. So now we can use this as a variable to let us uncover the biology and do it very cleanly. It's something that gets me excited. And I think we're really, honestly, it's early days for us in having these discussions with people around how should we best leverage these in panel design. So we've gone now to 7 labels in the blue, really five colors if you think about it, but I think a brand new dimension when we think around panel design. The next laser, which obviously we've been I'd say somewhat obsessed with is the yellow laser, right? And I think a lot of this comes from, honestly, the history of phycoerythrin. And, you know, we'd say PE, we say phycoerythrin, but like, I honestly didn't know what the history of phycoerythrin was. Turns out that the developer of the ultracentrifuge and his research assistant both had summer homes on the west side of coast of Sweden. And the way they used to extract this after the extraction protocol came out in 1910 is they would go out and get 44 pounds of red algae, bring it back, wash it with rainwater, and then start their protein extraction. That sounds pretty, honestly, somewhat beautiful, but also pretty laborious. We don't have to do that kind of thing in our lab now. But remember, too, that this has been really a foundational dye in our field, really thanks to its quantum efficiency. But it's been with us for a very, very long time, right? What is the history, though, of PE? First described use in the 80s, which is when it shows up here. Conjugation protocol comes out from the Herzenberg laboratory in the mid-80s. And then we get really the building of all of the different PE conjugates on top of this, you know, really that continues through to this day. We came out with 3 labels saying, look, we can create labels that are not cross-excited off the blue, where you can begin actually leveraging all of these laser lines. And then we've continued that story, thinking more broadly around what do all the PE conjugates that people use now, and how can we go in there and replace those so we can leverage all of the abilities of the yellow laser and everything else, right? So again, they're not designed to, you know, they're designed to be as part of a larger panel. You're also seeing that we're able to hit very small gaps, right? We're talking about 10 and 20 nanometer differences in terms of the emission here, which I think gets really exciting when we think about what the impact of that should be on hardware design in the future. So NovaYellow 590, really yet another, if you will, PE replacement, although there's a couple that could fit into that box. The yellows have a characteristic of, as I showed you, being really quite bright in comparison, for instance, to their blue cousins. It's compatible across a large number of different instruments because of its emission, very similar to PE. This actually is going to go through also a brightness increase. So you can think about this label being cleaner then, but also half the brightness of PE. So I think it's going to be a really interesting when we get into low antigen density markers. And you'll see that a number of the yellow labels are really bright when we get into separation index, but contribute less spread than something like a PE. NovaYellow 690 really targeted to hit and replace PE Sci 5 and Sci 5.5. If you've wrestled with these dyes or tried to use them in combination with something like PerCP Sci 5.5, I'm sorry, but the help is on the way, right? So we've got this 690 with a really good separation index. It does contribute spread. It's actually, in this case, lower than the other 2 comparator labels. But again, compatible with a large number of instruments that are meant to detect these 2 different tandems. 700 is another one. Actually, we're only 10 nanometers different. So in many cases, we're honestly, it's kind of the same story where we're going in and replacing PE Sci 5 and Sci 5.5. Again, depends on your choice and your panel setup, but now we're giving you an option that's only 10 nanometers different. 720, now we've stepped out 20 nanometers again. And again, we're actually, for some instruments, we're actually still in that same range as PE Sci 5 and Sci 5.5. So I guess the flyer would be, we need more detectors in that range. But again, a really interesting performing candidate where we can go in with less spread and really fit into a panel. So you've got a lot of options, for instance, if you're on a conventional cytometer, to then go in and replace those PE conjugates. There's a bunch of other reasons to replace those PE conjugates when it comes to stability, and just hold that thought for a moment. This is another one that's going through brightness tuning. So we expect to come out with something that will be almost nearly as bright as PE Sci 5.5, but have less spread contribution, right? Which I think is super exciting when we think about the ability to leverage that and the ability to leverage that with other blue dyes and other red dyes. So now we've gone to 7 labels in the yellow. We started with 4, right? Or started with 3 and added 4 on top of that. The next laser line to fall, if you will, to this thesis of, hey, we need to really go in there and think about what legacy dyes we have that have been great for where we started, but we need to head to a different place. As I mentioned, was APC, right? APC, for what it's worth, you know, allophycocyanin is actually literally a contraction of Greek words that means other algal blue protein, which I didn't know. That was news to me. It was first described in immunoassays in 84, and then it came out as part of the same protocol for conjugation from Randy Allen and Aaron in the mid 80s. And again, we've built a lot on top of this. We focused first on how can we go in and replace APC and APC Sci 5.5, and we'll be moving out further into replacing, you know, APC Sci 7. But again, with the same characteristics: low spread, very, very clean, clean emission. This joke for me never gets old, so I'm sorry to subject it to you, but these are our previous Nova Reds. We didn't have any. And now we're going in and we've actually got 4 off of the red. So you can see the types of emission that we're targeting here. Again, focusing it on APC, APC Sci 5.5. And again, able to hit some very narrow, if you will, emission deltas here, which I think is really interesting when we think around what multicolor panel design could look like when you've got clean fluorophores that can do that. NovaL 660, compatible across honestly every single instrument, because most are designed to detect APC. It is absolutely intended to replace APC. It is intended to be a better spread performer than APC. It is intended to emit cleanly in the red, right? And no, not holding back about its ability to stack up against APC. Similarly, it's been very interesting, again, a number of customer conversations where people have been trying to get access to APC Sci 5.5. It's either out of stock, you can't get it on mouse antibodies. We couldn't get access to it as well. So we really realized that there was a need for a replacement there, to hit something that you may have detectors for. NovaRed 685 is that label. Honestly, a very bright red label. You can see actually that compares very favorably to APC itself. Lower spread contribution than APC itself. And I think another really interesting dye that's also compatible across a huge number of instruments, right? Because of its gap to APC. Now we're in NovaRed 700. Again, very similar to that APC Sci 5.5 story, but yet, if you will, another option in there. Mostly though intended to go in and replace APC R700, right? Which does have a very large spread contribution. You can see its emission, for instance, here in the UV and the violet, but again, performs similarly on a separating basis. So you can have a label that's just as bright as APC R700, but contribute less spread. Another dye that's compatible across a huge number of instruments, and arguably you can see, you can even see it off of the Aurora detector, right? Arguably we don't have enough detectors off of the red, right? We only have 8 on this instrument. You saw earlier, we only had a handful off of the Symphony. Again, 710 and only 10 nanometers difference, so very similar to APC Sci 5.5. And again, actually in this case, a really interesting dye where we can actually get in and actually stack that space that APC Sci 5.5 was really intended to hit. Don't have anything for APC Sci 7 replacement yet, but that's in progress. So 4 brand new labels off of the red. So with that, maybe we'll pause, Seddon, if there's any questions, and then I'll hand it over to you.
Yeah, we don't have any questions thus far, but I'd like to encourage you to please jump in if you do have any questions. So I guess I'll go ahead and get started with some new applications, and feel free to interrupt me. All right. So if you want to go
help if I advance the slide, sorry.
Yeah, that's okay. All right. So sorry, there's always this moment where I have to move the things around on my screen so I can see everything. Sorry for the pause there. But what this is showing you is a panel, that you might use, say, if you had an attune. So, this would be also something that one could think about in terms of a Fortessa and really looking at this problem. So, why did we really start with looking at the blue laser and the yellow laser? And it's exactly this issue that you can see here. So, for instance, with PE dazzle, you can see that it would basically show up both in the BL2 and the YL2 in this conventional panel. And then you'd have the problem with PE Sci 5.5 and Percy-P Sci 5.5. Basically, the PE Sci 5.5 would bleed into the Percy-P Sci 5.5, so you wouldn't want to use them together. So this is just showing what you could use as, say, a T-cell panel and basically get these 2 new spots that, even though they exist on your cytometer, you can't really use them with current labels. So we do have a question. So the question is, how are we quantifying the spread, and is it relative to the dye class, or is it a strict scale? I can answer some of that. Mike can probably help answer some of that. It's a lot of math. So we did this on the Aurora, and we're actually looking at all, basically 64 channels, as it is on the Aurora. And I'm trying to think of the math, so maybe Mike.
So we're hand calculating spillover spread, Jack, and quite literally looking at it as, what does the spill look like into every non-primary detector? Is it relative to the dye class? No. Is it a strict scale? No. In the visualizations I was showing, we really pulled out, if you will, 5 bins of spillover performance. when you actually look at the spill of all of our labels, and then a number of different commercially available labels, right? Because we wanted to look at what the total universe, if you will, of spread looked like. Now, that being said, it gets very interesting on the Aurora, right? Because you do have so many detectors. In many ways, that's a very hard ruler to put up against your labels, but we like that, right? Because you really are looking at spread across a huge number of different detectors. And so you'll see spread contribution in anything that's not primary fluorescence, right?
Yeah, and one thing that we're also looking at just, going forward is also sort of setting up, because the Aurora has the ability to do something like virtual filters, also addressing that, for instance, for your individual instruments. So looking at, say, a typical, Fortessa or whatever, and we basically, since we have that, we can basically make those calculations as well. So it's been a question going forward, just so I'm fully transparent as we've been discussing that and getting those. So. All right. You want to go to the next slide?
Yeah.
All right. And so this is looking more at what you might have in terms of, a symphony type panel, and looking at what you are currently using now in terms of fluors and some of the issues that you might have, and what you might want to replace them with in terms of the NovaFluors. And you can see that there's multiple options going on, but there are things like, for instance, the BB660 and some of these other brilliant dyes, which are great in terms of brightness, but kind of problematic in terms of all the spread they introduce across the spectrum, as well as things like PE Sci5 and Sci5.5 that, are still, these issues, in terms of spreading everywhere. And again, the PE dazzle, the same kind of thing, spreading into this, the blue region. Just because you've got more channels with the symphony doesn't mean that you don't have, again, these sort of spread issues going forward. And so, yeah, so the last part of that was showing that, also there are these traditional issues with AP Sci7 and PE Sci7. And so this is looking specifically at APC Sci7. And what you can see, is, a well-known issue where there is the stability, and it's pretty fast, in terms of once you put this into fixation, you start seeing the Sci7 actually getting, removed. And so you can actually see this increase in brightness, as you're looking at the APC channel, because you're losing that Sci7 off of there in the fixative. And so that's obviously a problem when you're looking at, okay, is this actually showing up in Sci7, APC Sci7 or APC? You really don't want to lose your data and change what you're actually seeing. All right. And so one of the things that's been fun is that we've really looked at some of the stability issues. And one of the great things about the Phyton is that it really serves as this solubility cage. So in terms of stability, this is using, antibody conjugated, and this is, in this case, a 1 cluster Nova Yellow 610 to an anti-CD3 antibody. And what you can see is that at day 0, you have the same separation as you do when you fix this for 14 days and just leave it in the fixative. So that's really, surprising you're not introducing some kind of issue there. And then this was what I like to refer to this as the Europe experiment, because as we've been shipping everywhere, what you find is that, you think it's going to get through customs and it doesn't quite. And so, what we wanted to look at was, okay, well, what is happening if this doesn't get there and, is basically being left at room temperature for some period of time. So this is taking not just the fluors, but also the antibodies, and either leaving them, at 4 degrees, which is where you would typically store them, or at room temperature, and then staining cells over time. And what you can see is that even if you leave these labels out, on the bench with the antibodies, basically you have the same staining. So it's really great knowing how stable they are in terms of, being able to use them. And if there are these issues, it's not a big concern.
Yeah, and so we've got some questions, and I think that maybe we use this slide to sort of tee up, which is compatibility with brilliant buffers, with DNAs, and with serum,
Yes, yes, absolutely. So when you think about blocking, there's really a lot of different things that we think about blocking. So there's the traditional, okay, you know, and more so on the mouse or, you know, monocyte or you know, more granular populations where you have the, you know, higher expression of the FC receptors. So that's going to be blocked with something like an FC block, or, you know, in the mouse 2.4 G2. And then you also have, you know, the blocking with the brilliant dyes, and that's actually based upon the fact that you can get these polymers forming. And so you know, we also have a third option, which is what we refer to as NovaBlock. And so you can use NovaBlock in conjunction with these other with these other blocks. And basically, you only need to add the NovaBlock 5 minutes before you do the staining. And you can use this in conjunction with the brilliant buffers, or you know, any other, say if you want to use serum or FC block, any of these things. So let me show you. Okay. I don't know if that's showing it for anyone else, sorry. Okay, there we go. Yeah. So what you can see here is that this is actually showing our NovaBlock and comparing it with either NoBlock or BioLegend TrueStain. And that's actually abbreviated. That's the BioLegend monocyte TrueStain. And so one of the issues that's known about the monocyte population is that it can actually have some non-specific interaction with the labels themselves. So things like, you know, some of the P tandems, some of the side eyes. So this is just showing you that what you can see here is, again, when you have no block, you actually have this increase in the background. So what, you know, it's not changing your overall fluorescence, but that increase in the background is of course going to really change your staining index. So really a problem. And what you see with our NovaBlock, or again, similarly, the BioLegend monocyte TrueStain, is that it reduces this background and therefore you regain that good separation index that you have. So if we go on to the next slide, you can see that this is actually very specifically happening for the monocytes. So on the x-axis, you have CD14 and APC, and then the different Psi or, you know, other PE tandem dyes. And you can see that specifically this background is popping up in the monocyte type population. And this is also true for some of our NovaFluors. And so that's why using NovaBlock, especially when you're looking at these monocyte type populations, is important. And we do include that in both our CD4 kits and our conjugation kits, and also have the ability to sell that separately. So. All right, so this is, you know, fun because it's really cutting edge data. And so what I did here was actually, we've been asked a number of times, you know, well, what about using this intracellularly? And we, you know, have said quite honestly, we have to test it. So let's see what we get. And so what you can see here is I have, you know, human PBMCs that have been stimulated with PMA ionomycin. So for the first hour, they're just stimulated with PMA ionomycin, and then they have an additional 5 hours where the Berfeldin A is added to block up the Golgi secretion of, in this case, you know, interferon gamma. So what you can see here, I have the control at top where you can see that 18.4% of the cells are producing interferon gamma. And then when you look below, in terms of this unstained, we used interferon gamma with Nova Yellow 660. And again, this is being very transparent in terms of that data. So you can see this increase in, you know, as we're adding more of the antibody with the Phyton. So the first answer is we're definitely getting into the cells, and that does not seem to be an issue. So that's really great. The other interesting thing that you can see is that it seems like there are two populations. One that seems, you know, visually to be the actual positive population. And then as you go further, this creeping background that you see. So overall, as a first experiment, I was really encouraged to see that we are getting into the cells. And I think we really just need to work on this background issue. And we have a number of experiments planned and some controls that we need to run to really see like where that background is coming from and how we can block that up. But as a first run, I think it was very encouraging.
Yeah, it's exciting. And it's worth mentioning, right, that the size of the Phyton is 150 kilodaltons and it's about 20 nanometers across if you lay it flat. So it did keep coming up as a question, right? It was smaller than PE, right? And we saw the stability of it in fixative. So we were feeling optimistic, but it's more important that we look at this and yeah, to set the point, optimize what it's going to look like in the cytoplasm, what it's going to look like, and then try and take that further into the nucleus. Cool. Thank you, Sid.
Yeah.
So how does all this, like, how do we get access to this? And also, I think the question that we've been asked a number of different times is, can I please get conjugated antibodies? And so we'll talk about that. We'll also talk about what are customers telling us about the conjugation kit as well, because it's something that we've spent a good amount of time thinking about and attempting to make easy as well. Again, so I'm going to bring back our launch paradigm, right? Which is, how do you stage a launch and how do you think about reusable components? And that's exactly that. And yes, we are going to talk about conjugated antibodies, and I'll just say it again. So the way that we like to think about a launch is actually in the following way. For all new labels, it's always enabling a CD4 kit for human and for mouse, right? And that in many ways is the reusable component, because that's what enables you to do panel design. It also is what enables you to characterize the labels for yourself, right? We're definitely, setting our big, don't take our word for it type people. And so, we've got a number of these kits out for people to take a look at. Well, what is the spillover and how does it work across a set of instruments? The second stage, if you will, of this is always conjugation kits. We will always do that as conjugation kits for new labels. It gives you access to put this on content, right? It gets you away from having to, wrestle with the timelining or the difficulties of doing a custom program. And I'll also mention that, while, conjugation kits sometimes are very challenging, I'll actually show you what our customer reviews look like of ours, because we focused on making that easy to use and very fast to use. So hold that thought for a moment. And then all labels, of course, as you'd expect, are going to mature up into both a customs program, which we're enabling milligram and up conjugations of antibodies, but also our own conjugated content. So we'll describe what that means and what does that look like even on timeline. So what we've done is we have a large number of customers using CD4 kits, also using conjugation kits for these first 6 labels, right? And I'll show you what the feedback has been on them in a moment. We're going to move those up, if you will, into our own conjugated antibodies, and also a customs program with late July shipping. So I'll show you what those antibodies look like in a moment. We've done it across both mouse and human, and so I'll show you that. We'll be doing CD4 across all of the different markers that we offer, as you'd expect, really for testing. And all of those are available for pre-order actually now, and then we're doing it in late July shipping. All of the labels that I've described today, all right, all 13 new floors, obviously rolling up into CD4 kits and conjugation kits. Late September shipping on these, actually staging that, right? And so we're working with a number of people who want to get access to new labels. We'll actually stage that and actually ship them to you as they're ready. Again, I think it's really important we have this reuse component of CD4 testing for panel design or for characterization, as well as a conjugation kit. We literally just got these reviews this morning, which is from our conjugation kit, right? And so we worked very hard to make the conjugation kit, one, easy to use, right? We're talking about 5 pipetting steps, no special equipment needed, right? You just need a centrifuge and a heat block. And, we got these out and got these unsolicited reviews back, right? Which is, I was super excited to see this, 5-star rating across all, particularly on after sales service, which is totally a credit to Set in. She's been very focused in on making sure customers are successful with both the conjugation and implementing this into their panels and making the point around how you can stack this. And then another customer, literally just posted up before this webinar, that this was easy to use, less spillover in the performance that we're seeing. So again, don't take our word for it. These are customers who now have access to this, and you're using this right now in their work and are really excited about the ability to even conjugate these, right? And leverage it, and within a day have new colors they can leverage on their flow cytometry experiments, which honestly is it's super exciting to see this. But we know conjugated content, right? There's a big desire for conjugated antibodies. And so what we did is we took a little bit of a different approach, because we've been talking around, okay, well, we've got these clean fluorophores. What should we do to do this really intelligently? And so what we did is we designed 2 different sets of specificities, right? We called it human 1 and TBNK. and thought around, okay, well, what do we want to do with those? We've got a mouse panel as well that we want a certain number of specificities. And what we did is we actually pulled from the CD maps database as well as publicly accessible data on the mouse, and actually looked and mapped this to antigen density. And so we created, essentially, a deterministic way of looking at, well, which color should we offer these on? And what we did, instead of optimizing based on the old heuristics of panel design, which are all about dealing with the compromises of spillover and dealing with the compromises of spectral, these very large promiscuous emission peaks, we actually optimized based on the separation index, which is the ability to actually pull apart the biological populations, and spillover spreading. And so that's actually how we went and chose what colors to put what antibodies on. And the goal there is actually to provide a set of phenotyping markers that, again, will enable you to phenotype these populations very cleanly and with the lowest amount of spread, which I think is important because, as you can appreciate, a lot of these are also high antigen density markers. So with that, these are the set of 12 antibodies that we're putting out. There's some very familiar clones, I'm sure, to you if you're in the human or non-human primate world. Essentially, there's a swap out into this NovaYellow 570 where you can use it on either CD16 or CD8, as actually the antigen densities there are pretty similar. And then on mouse, we're coming out with a set of antibodies as well. Again, some clones that should be very familiar to you. Each one of these has been optimized, again, for spillover spreading and having that characteristic of having high separation with low spill, right? And so you're getting that, if you will, faith that you've already gotten that optimization. We obviously need to build out the menu from there, but we've got to start somewhere, and this is where we're starting. So again, all of these will be shipping in late July. We're pretty excited, honestly, to get this into some of the custom projects that we've taken on into the hands of customers and see how they do. We've launched pre-order quote requests for both the conjugation kits and the CD4 kits on our website for the new labels. I'll be rolling up that conjugated content and the conjugated antibodies very, very soon. So that way that'll be searchable and you can take a look at that. Eventually, we'd also like to share some of the more interesting thoughts I think we have around panel design and how can you do that when you do have a very large number of clean fluorophores. And it's been exciting, honestly, to just see people, if you will, pulling in these new floors and really signing up, if you will, for what, hopefully we think of as sort of a new era in fluorescence. What's next? How does this all look, right? And so, now that you've got 19 floors on these lasers, I think it's pretty obvious that we'll be building out into the larger, the longer wavelengths in the blue and the yellow, and particularly in the red, to go and replace something like APC size 7. But I think, the next place to go is pretty obvious when you consider what 5 laser lines we have. And the answer is going to be, obviously, there's going to be some purple rain. And so, I think hopefully we've given you a sense of the pace and the way that we like to think around designing these floors, right, with low spread and compatibility across all these detectors. And if anything, I think it's going to be very interesting to put the pressure, if you will, back on hardware and say, we need hardware that's got narrower detection, so we can detect these things that could be 10 or 20 nanometers across. What's also next is we will take all of our labels and move them up into conjugated content, right? As you'd expect, we're engaging with a number of different people to figure out how best to do that and how soon to do that. I think we've just found that honestly, it's honestly in many ways faster for us to serve customers directly. And then, we'll be moving up new colors in through the exact same launch, if you will, protocol that you've seen here. Obviously, I think one of the things we're doing very actively is now taking this and now pushing the number of parameters that you can do in multicolor flow cytometry, right? Be that on a conventional cytometer or be that on a spectral flow cytometer, right? We're going to be pushing very, very hard on that. We've done it once at 40 colors, showed how clean we can do that with our floors. You can expect that we will be taking these labels with extreme alacrity and trying to do a very large experiment and really teeing that up for Cyto and sharing that data again, as we always have. I would also argue, clean is the new bright, right? You want to have faith that the markers you're pulling out on populations are really there. Had some great conversations about that, right? In terms of what does removing away from spread mean? Expanding conjugated antibodies, pretty obvious one. We'll be going for full vertical on colors. Again, we've relaunched 19 in the first half of this year. We still got a little bit of 2020 left. And then also expanding the applications of use. I mean, I think there's a lot of great questions around, can you do phosphostats? What does this look like in immunofluorescence? And if there are applications that we haven't covered today or haven't mentioned, or the ones I just did, please reach out to us. And I think, we're very much in a collaborative mode where we know what we can do and what we can't. So anyway, I think hopefully you share my excitement around having these kind of performance on fluorophores, having them, if you will, this quickly. And I would just note, here's our contact information. This is my email address, Mike Stedniski at Phitonex.com. Info@Phitonex.com actually gets set in ORI. or you can email her directly because you see your name there. But all the pre-order requests are launched for CD4 and conjugation kits. We're, again, rolling up some of this conjugated content soon, or you can just send me a quick message and say, hey, I'd either like to learn more or I would like to check these out and get these into my lab. So, yeah, I see we've got some questions, which is great. Keep them coming, and we'll hang out and hopefully have a good conversation.
Yeah. So one of the questions is, do we see a change in the fluorescent spectrum when staining on cells versus beads? And so I've been doing a lot more of the work on that. So you can actually, I mean, obviously there's the sort of autofluorescence that you see on, even on PBMCs, somewhat into the UV and the violet. For the most part, we haven't seen that, but we're really interested in looking closer at that. And then we've also, not only are looking at the current beads, but also there's a new set of UltraComp Plus beads, which are supposed to affect, I think, as there's more of a shift into the red. So I've got sort of a lot of questions that I'm really trying to look at, and that's actually, I'm going to be running that new set of beads and comparing all 3 sets tomorrow. So yes, a question I'm certainly interested in. And then we have another question from Christian. So how do these fluors work when using tissue samples where DNA has been added to the sample prep, and also tissues that have low marker expression like the brain? So I can start by answering one of those. Is that, we would, first off, generally it's good, to wash out some of the DNase. The phyton itself is very resistant to DNase. I think if you had a high amount of DNase, there could be some, issues in terms of the linkage between the antibody and otherwise. But I think there's some data Mike, has on that we have on basically, as long as it's not such a high amount of DNase, that it's not an issue there.
Yeah, the phyton itself is resistant. It can't actually be recognized by the nuclease, so it doesn't bind on there, right? And so, but yeah, we've, if you wash out the DNase post essentially tissue treatment, right? It won't. You don't have enough in there to cleave off the double-stranded DNA linkage you've got to an antibody, right? Because those conjugation kits, particularly, you're activating the antibody, you're putting a single-stranded oligo on them, and then you've got the complement hanging off of the phyton, and that's how that structure is constructed. And so, at high DNase concentration, I mean, you will certainly cleave off that double-stranded component, right? You can see that as a disadvantage, or you could see it as an advantage, right? Because what you can also do now is you have a very simple system for sequential staining. I'll have to leave it there.
Yeah, we have not gotten into a tissue like the brain. So again, those just to some extent, I think our access to mice, probably like almost everyone here, has been somewhat delayed by COVID. So, we're able to get, frozen splenocytes, but some of the experiments we'd like to do have been a bit delayed in that regard.
Yeah, so a totally relevant and question that we dealt with in the, that we thought a lot around in the spring, and I'll take it on, which is how does your pricing structure compare with those of your competitors? All too often, such novel reagents are way too expensive for many of our researchers to use. Thanks. First of all, totally agree. And so what we've done is we actually price the CD4 kits exactly the same way on a per test basis that you can buy CD4 kit, some of the testing reagents from BioLegend. So they are priced exactly the same as those testing volumes. So that's the testing part. The conjugation kits, those are, not only are they competitively priced as a conjugation kit, one of the things we did when we were really rolling out these to break out the colors is that arguably they're very competitive with conjugated antibodies. Why do I say that? A 1 by 100 microgram conjugation kit for any color is $460 US. But that's for 100 micrograms. That's not for 100 tests. That's pre-titration. So I would argue that on a per test basis, after you titrate, that's very competitive with what you're doing, right? For have these novel reagents and actually have them be on a per test basis, very competitive with what you're doing. And the final part of this, of course, is conjugated antibodies. These are priced exactly the same as what you would expect to pay for other conjugated antibodies that are coming off of, if you will, sort of higher end dyes, right? They're not priced above that. So the mouse antibodies are $345 US, all of them. And then the human antibodies, I think, are $395 US, right? And do we offer a degree granting discount? The answer is yes. We can't do that right now on the CD4 kits, but we offer it for other things. So this was definitely I would say an issue that a lot of customers actually brought to us, and they're like, look, I really want to use this, but, and so that's how we've attempted to address it. I would also say, you know, our prices are all up on the website as well. So we try to be really open around, this is how they're priced. And if you've got any detailed questions on that or how we like to think about that, I'm happy to discuss further, because that's something that was honestly a great amount of customer feedback we had early on, to be able to really get, to your point, to the question's point, get research access to it. Oh, one final one I didn't mention is block. Block's actually priced underneath what you would pay for other blocking solutions. So just another note. Yeah.
All right, great. Do we have any other questions? Let's see. All right. Well, thank you very much to Mike and to Seddon. We really appreciate it. It was a really great talk. And for those of you who may have missed the early part of the talk, we will have this recording up at the metroflow.org website in just a few days. So you can check back and reference back to anything you may want to see again.
Thanks all for attending.